Good morning, students. Today we will study about pollen pistil interaction. Fine. And this is a very important step in the process of fertilization in angiosperms. And this interaction includes some events, like first one is recognition of compatible pollen grains. Second is growth of a pollen tube, and third is the entry of pollen tube into the ovule. So, all these events from the deposition of pollens on stigma till the entry of pollen tube into the ovule are collectively known as pollen pistil interaction. Fine. Now, let us discuss these points individually. The first one is recognition of compatible pollen. Fine. See, these are the pollen grains and this is the stigma. Fine. Style, ovary, ovule, and inside the ovule this structure is embryo sac, or you can say the female gametophyte. And embryo sac contains 3 antipodals, 2 polar nuclei, polar nucleus you can say, or 2 polar nuclei, 2 synergids, and the one is egg cell. Okay. So, this is embryo sac. Now, first is this step. When these pollen land on the stigma, then immediately pollen tube will not arise. Why? Because first stigma will confirm that these pollen grains are compatible or not. मतलब वो पहचानेगी पहले पोलन ग्रेन्स को कि पोलन ग्रेन राइट टाइप का है या नहीं है. This pistil, has the ability to recognize and accept the right pollen of same species and to reject the pollen grains that are incompatible either of same species or of the other species. And how this pistil or stigma will recognize the compatible pollen grains? Actually, there will be an interaction between the chemicals released by stigma as well as the pollen grains. If that reaction will be successful, then obviously what it means, that stigma has accepted the pollen grains, means the pollen grains are compatible. अगर इन दोनों के बीच की इंटरैक्शन सक्सेसफुल हो जाती है, दैट मीन्स स्टिग्मा ने पोलन ग्रेन्स को एक्सेप्ट कर लिया है. फाइन. So, this is the first step, that is the recognition of compatible pollen. Second step is the growth of pollen tube. Now, after accepting the compatible pollen, what happens? These pollen grains will absorb water and they will get swell. Fine. The pollen grains germinate and they will release the pollen tube. Now, pollen grains contain vegetative cell and the generative cell. We know that generative cell will further divide into 2 male gametes. Fine. So, what happens? This pollen tube will initially, this will initially take the nourishment from the pollen grains, because pollen grains contain what? Vegetative cell, which contains abundant amount of the food. So, initially the pollen tube will take the nourishment from the pollen grains, and after that this pollen tube will take the nourishment from the interior of stigma as well as the style. Fine. अब ये pollen tube आगे बढ़ेगी and these pollen grains will pour its content into the pollen tube. Fine. अब हमने पहले पढ़ा था that pollen grains in 60% angiosperms shed at 2 cell stage and in 30 or you can say the 40% angiosperms shedding takes place at 3 cell stage. 2 cell stage मतलब यहाँ आने तक पोलन ग्रेन्स के अंदर 2 सेल्स होंगी, वेजिटेटिव और जनरेटिव। बाद में जनरेटिव सेल डिवाइड होकर और 2 मेल गैमेट्स प्रोड्यूस करेगी। लेकिन 3 सेल स्टेज का मतलब क्या है, बिगिनिंग से ही पोलन ग्रेन्स के पास 3 सेल्स हैं, दैट इज 2 मेल गैमेट्स एंड 1 इज द generative cell. Fine. So now pollen tube will move. These pollen grains will not move. These pollen grains will pass their contents into the pollen tube. Now pollen tube will move further. This pollen tube will secrete some enzymes and those enzymes will digest stigma as well as the style. Fine. Third is the entry of pollen tube into the ovule. अब pollen tube यहाँ तक पहुँच चुकी है. This is the structure of a ovule. See, this is the pollen tube. Fine. Pollen tube may enter through the micropylar end and it is called as porogamy. This pollen tube may enters through callosal end called as callosogamy, and this pollen tube may enter through the integments, these integments, coverings, internal as well as the external integments called as mesogamy. So entry of ovule through micropylar end is porogamy, through callosal end is callosogamy, through integments is called as mesogamy. These two are very rare. Porogamy is very common. Fine. So pollen tube will enter through the micropylar end, and here you can see these are the finger like structures. These finger like structures are called as filiform apparatus made by the synergids. We know that 2 synergids are present over here. These 2 synergids will make this filiform apparatus. Fine. And the function of this filiform apparatus is this structure will release some chemicals and direct the entry of pollen tube. Means it will guide the entry of pollen tube कि जाना कहाँ है. Okay. And one of the synergids will degenerate. See, only one synergid is there. Where is other? Other synergid will get degenerate. Why? Because जब pollen tube enter करती है और वो burst होती है, bursting का जो impact है, उस impact से एक synergid पहले ही degenerate हो जाएगा, और जब pollen tube अपना contents pour करेगी यहाँ पर, उसके बाद सेकेंड वाला synergid भी degenerate हो जाएगा. फाइन. सो सिनर्जिड्स का फंक्शन क्या हुआ, ये पोलन ट्यूब की एंट्री में हेल्प करते हैं. जैसे ही पोलन ट्यूब अपना सारा कंटेंट यहाँ पोर करेगी, सेकेंड वाला सिनर्जिड जो होगा वो भी क्या हो जाएगा, डीजनरेट हो जाएगा. फाइन. अब ये स्ट्रक्चर किसका है, एम्ब्रियो सैक का है. ये जो एंटीपोडल्स हैं इनका क्या होगा, दीज एंटीपोडल्स आर द वेजिटेटिव सेल्स ऑफ एम्ब्रियो सैक, not the pollen grains. These vegetative cell or the nucleus having abundant food, these are of pollen grains. Fine. And these antipodals are the vegetative cells of embryo sac or the female gametophyte. Fine. And they will also degenerate very soon, or you can say sometimes they absorb the nourishment from the nuclear cells. Nuclear cells are a mass of parenchymatous cells present inside the ovule. Fine. So this was all about the pollen pistil interaction. All these events, these 3 events starting from the recognition of compatible pollen till the entry of pollen tube into the ovule are collectively known as pollen pistil interaction. Okay.